Right, class. So, we talked about two types of ligands, right? We have acetylcholine. Well, we have many types of ligands, but we are focusing on either acetylcholine or norepinephrine and epinephrine. Acetylcholine binds to specific receptors, and those are called cholinergic. Norepinephrine and epinephrine bind to adrenergic receptors. There are two types of cholinergic receptors, nicotinic and muscarinic. Okay, nicotinic is always excitatory. So it excites the downstream cell. The downstream cell can be another neuron or muscle. So it's always excitatory. Muscarinic can be excitatory or inhibitory. That's muscarinic. Okay, what else do we need to know? The nicotinic receptors, obviously the ligand needs to bind to the receptor, and that open up the gates, right? In case of muscarinic, the ligand binds to receptor, and the receptor is a G protein, is coupled to a G protein. So in case of muscarinic, we have G protein coupled receptors. Okay, where in the body do we find nicotinic receptors? Well, we find nicotinic receptors in the central nervous system, in the peripheral nervous system, and also we find it on skeletal muscles. We find muscarinic receptors on the central nervous system. We find it on the heart. We find it on smooth muscles. We find it on the pancreas, GI tract, etc. Okay? All right. And then we will talk a little later about the adrenergic receptors. Now, moving on to the voltage-gated channels. Voltage-gated channels, obviously, remember, they'll require some sort of electrical current to open. And there are three types that we will talk about. The first one is voltage-gated sodium channels. And we have learned about voltage-gated sodium channels during our lecture on muscles. Then we have voltage-gated potassium channels. And we also have voltage-gated calcium channels. Let's talk about each of these voltage-gated channels. Okay, to start with, let's talk about voltage-gated sodium channels. Voltage-gated sodium channels. First, voltage-gated sodium channels open at threshold. Do you remember threshold on the number line that I drew? My negative 55 is threshold. So at threshold, which is negative 55, we have these channels open. As voltage-gated sodium channels open, which way do the sodium move? Well, from outside to inside. So as they open, sodium moves in. And as sodium moves in, that brings in positive charge inside. So the inside becomes less negative, more positive, right? And that leads to depolarization. Depolarization. Okay. And when do these voltage-gated sodium channels get deactivated? Well, it becomes positive. So the membrane polarity changes to less negative, less negative, less negative, less negative to 0. Ultimately, it moves to the positive side, and these channels close at positive 30 millivolts. That's when these channels close at positive 30. Really important to remember. And I want you to kind of memorize these, and this will help us going over the action potential. All right. The next channel that we will talk about, those are called voltage gated potassium channels. When do they open? They open at positive 30 millivolts. That's when they open. Okay. As they open, what happens to sodium? What happens to potassium? Does potassium move in or out? Potassium rushes out. As potassium goes out, are we losing positive charge? Yes. So due to that, the inside becomes more negative, less positive, a condition called repolarization. So the cell is moving back to repolarized state, meaning it's coming back to resting membrane potential. And then during our muscles lecture, we learned about voltage gated calcium channels. And for testing purpose, just to know that they open they are present in the axon terminal. They're present in the axon terminal, and of course they open in response to action potential. Okay, so let's review this. We have 3 types of voltage channels. We have voltage gated sodium, voltage gated potassium, and voltage gated calcium channels. When do these voltage gated sodium channels open? Well, at threshold, meaning negative 55, voltage gated sodium channels open. When sodium moves in, as sodium moves in, the membrane potential becomes less negative, more positive, a state called depolarization. And it continues to depolarize until it hits positive 30 millivolts. And at positive 30, these voltage gated sodium channels close. Okay. By close, when I say close, are they close or are they deactivated? At positive 30, they reach deactivated state at positive 30. So let's put it here. So at this, when it reaches positive 30, voltage gated sodium channels are deactivated. You must activated. You must remember that. Okay. They are not closed. They are deactivated. Okay. Now, let's talk about voltage gated potassium channels. They open at positive 30. And as they open, potassium leaves the cell, making inside more negative, less positive, and that brings everything back to resting membrane potential by the process of repolarization. And voltage gated calcium channel, they're present in the axon terminal, and they open in response to some action potential. I want you to see one thing. See, the opening of the voltage gated sodium channels and the voltage gated potassium channels are different. Sodium channels open at negative 55, potassium channels open at 30, positive 30. So they do not open and close together. Sodium opens first and then it closes first. When sodium closes, then at positive 30, the potassium channels open and they close later. Okay, and we will see all these when we draw out the action potential graph. Okay, so this is again just a review slide from what we have drawn out. talking about the sodium channels, ligand-gated sodium channels. So we need a ligand to bind to a receptor which will open up the gates, and as the gates open, sodium will rush in. So the ligand is acetylcholine, and the receptors to which acetylcholine binds, those are called cholinergic receptor. We did talk about that. There are two types of cholinergic receptors. We have nicotinic one, which is always and always excitatory, meaning it excites the downstream neuron or muscle, or it can be muscarinic, which can be either excitatory or inhibitory. We also mentioned that muscarinic receptors they use G protein and second messenger pathways. You must remember this. So where do we find acetylcholine receptors? We find acetylcholine receptors in the CNS, in the CNS, we have both muscarinic and nicotinic receptors. In the autonomic nervous system, we have both muscarinic and nicotinic receptors. And in the neuromuscular junction, we only have nicotinic receptors. All right. We also talked about the sodium voltage-gated channels, potassium voltage-gated channels, and calcium voltage-gated channels. So I'm not spending more time on this. Okay, this is again a review slide. So the voltage-gated sodium channels, we will start with the picture on the right-hand side. So we see that in this state, the channel is closed. Right? Enclosed, it's closed and the ball is kind of hanging. Then when the channel is open, everything can move. And then in inactivated state, the channel is closed by the ball. Okay, I'll stop here and then start with a new video.